Recall that chromosomes are comprised of chromatin, and that word explains how it is a complex of DNA plus proteins. Chromatin describes the organizational structure. Now, early scientists were able to recognize that chromosomes were inherited and chromosomes carried the genetic instructions of life. But what part of chromatin? Was it the DNA or the protein? The discovery of the genetic role of DNA began with the research by Frederick Griffith in 1928. Griffith worked with two strains of bacteria. One was pathogenic and one was harmless. When he mixed heat-killed remains of the pathogenic strain with living cells of the harmless strain, some living cells became pathogenic. They were able to take in genetic information. So in 1952, Alfred Hershey and Martha Chase performed experiments to determine that it was DNA, not protein, that was the one component that was able to cross in to the cell. To do so, they designed an experiment where they tagged either protein or DNA. They built bacteriophages with this radioactively tagged protein or DNA, and then allowed those bacteriophages to infect bacteria. And then they examined the bacteria to see whether they took up radioactive proteins or radioactive DNA. And they found only DNA entered the bacterial cell. So now that we knew DNA was the genetic material, we can examine it more for its structure and utility. Know the hallmarks of DNA, which stands for deoxyribonucleic acid. The DNA molecule consists of two polynucleotide strands that spiral twisted around each other. We call this a double helix. These two strands are anti-parallel, meaning they run in opposite directions, and they are held together by hydrogen bonds. The nucleotides come in two forms, purines and pyrimidines. The purines are double-ringed, and the pyrimidines are single-ringed. It was recognized early on that replication would involve copying information from the parent strand to create new daughter strand or newly synthesized DNA, suggesting some sort of complementary or template mechanism to fill in the gaps. There were several hypothesized models. A conservative model would suggest that the parents separated, served as templates, and then rejoined. A semi-conservative model would have this parent strand separate and each serve as a template for the new strands, which would be composed of both old and new. And a dispersive, which would chop up the DNA into a number of segments and copy different pieces. So each resulting daughter strand would be a mix of old and new for many generations to come. Messels and Stahl conducted their famous experiment on DNA replication using E. coli bacteria as a model system, and they set out to determine which of those models was the model. And so they hypothesized by using parent DNA strands that were tagged blue and daughter DNA strands that were tagged red, they would be able to discern conservative, semi-conservative, or dispersive patterns based upon the resulting color of subsequent DNA copies. What they found was in the first generation, there was a blue strand indicating that the parent DNA had been made correctly. Replication after that, it was a purple strand. And in the third replication, they had purple and red, suggesting that the second copy was a mix of blue and red. And after that, all subsequent copies contained only the new red nucleotide. It begins at an origin of replication. This is where the two DNA strands are separated, creating what's known as a replication bubble. A eukaryotic chromosome may have hundreds of these, and replication will proceed in both directions until the entire molecule is copied.
DNA replication begins at specific chromosomal sites called replication origins. At the origin, the DNA duplex is melted by specialized proteins, resulting in a replication bubble. DNA synthesis starts at the origin and proceeds bidirectionally. Two replication forks move outward in opposite directions. At each growing replication fork, 2 new DNA strands are made. The leading strand is synthesized continuously from a single RNA primer. It grows in the 5' to 3' direction, the direction of replication fork movement. The lagging strand is copied discontinuously from multiple primers, resulting in a succession of Okazaki fragments. Although the direction of DNA synthesis is opposite replication fork movement, the overall direction of lagging strand growth is toward the replication fork. As replication proceeds, the RNA primer of the first Okazaki fragment is replaced by DNA. Then the lagging strand fragments are ligated together.
So at the end of each replication bubble, a replication fork, a Y-shaped region is formed. This is where the new DNA strands will be elongating. Enzymes such as helicase, topoisomerase will assist. The helicase untwists and separates the DNA double helix. Topoisomerase corrects overwinding at the other side of the replication fork. Once open, single-stranded binding proteins will bind and stabilize the single-stranded DNA so they don't collapse in and reseal. Next in comes primase, an enzyme that can start an RNA chain from scratch. It will lay down a small runway of RNA nucleotides using the DNA template. This is known as the primer. It is the starting point for DNA elongation. DNA polymerase can attach to the primer and add nucleotides. It can move really fast, roughly 500 nucleotides per second in bacteria and 50 per second in human cells. Finally, the enzyme ligase is used to join any 2 fragments of DNA together, where these origin of replications fuse into one. In terms of visualizing this process, we often describe the replication complex as moving down the strand, a train on the train tracks of our DNA, when in reality, it's a stationary complex that reels in the DNA. How do we make sure that the DNA is being copied with high fidelity, error-free? We can't. Given the size and speed, the more bases you have, the longer it'll take. But time is of the essence because cell replication creates a very vulnerable state for these cells to be in. And for the DNA to be copied and exposed, the longer that takes, the more likely damage might occur. So there's a trade-off here. The copying of DNA must be remarkable in its speed, and there must be mechanisms in place to help proofread to ensure accuracy. It's not perfect, but the error rate is small. An error roughly 1 in every 10,000 bases. Let's examine 2 of the proofreading processes. The first is mismatch repair. This occurs during DNA synthesis. It involves endonucleases, enzymes that cut the phosphodiester bond within the nucleotide chain. That gap is then filled by DNA polymerase. It is a precision replacement mechanism. Contrast this to nucleotide excision repair. This occurs whenever damage happens to the DNA after duplication. Damage from just life, chemical or physical agents, like a sunburn. Nucleotide excision repair involves exonucleases. These are enzymes that cleave from the end and they remove a large DNA fragment from the outside of one of the DNA sequences. That gap is then filled in by DNA polymerase 1, cleaning out the whole stretch. If DNA duplication has rare errors, 1 in every 10,000 bases, and the error of the proofreading mechanism is also low, another 1 in 10,000 bases, that means roughly 1 mistake happens every 10 million bases, but it's not 0. And so while humans are 99.9% identical, given that 3 billion nucleotide genome, there's still roughly 3 million differences accounted for through this random error and mutagenesis. But this isn't necessarily a bad thing. Remember that evolution occurs when there are sequence changes, when these changes are passed on to the next generation. And these differences allow us to identify unique individuals and circumstances. Chargaff's studies on DNA structure, specifically the anti-parallel nature of DNA, gave rise to two particular rules. The first one is that the base composition of DNA will vary between species. The percent of T's versus A's versus G's versus C's would be unique to humans or monkeys or mice, and that all organisms within that species would be relatively similar. But between species, there would be distinct differences. Chargaff's second rule was that the number of A's would always equal the number of T's, and the number of C's always equal to the number of G's. We would be consistently pairing a purine base with a pyrimidine base. And this made sense. Because if you could pair any two nucleotides together, how could you have a template strand? And then, given the size differences between a purine and a pyrimidine, your DNA might be 4 rings wide or only 2 rings wide. That inconsistency would make it liable for damage, nicks, improper packing. So by consistently pairing adenine to thymine, cytosine to guanine, we were consistently getting a purine and pyrimidine pair, so the DNA helix was always 3 rings wide. At the replication fork, DNA elongation is happening in 2 directions, creating 2 different daughter strands. One is known as the leading strand, the other is known as the lagging strand, because it's being elongated in small segments that will then need the additional step of ligase to join them together. When describing DNA, we coin one strand the leading strand and the other strand the lagging strand. Let's see this more in detail.
In this image, I labeled the top original strand 3' to 5'. I labeled this bottom original strand 5' to 3'. That's the original DNA that is going to be replicated. DNA is unwinding here thanks to helicase. In this example, it will keep unwinding in this direction. Primase places primers. DNA polymerase is building the new strands. Now, the thing about DNA polymerase is, when it's building a new strand, it can only build the new strand in the 5' to 3' direction, meaning it adds new bases to the 3' end on the new strand. See how it's being built in the 5' to 3' direction? This one is called the leading strand. But take a look down here. So DNA polymerase once again is building a new strand in the 5' to 3' direction. But there's a bit of a problem here. See, as DNA unwinds, because DNA polymerase can only build the new strand in the 5' to 3' direction, it has to keep racing up here next to where this unwinding is happening. You can see why then this new strand is known as the lagging strand. On this lagging strand, primers have to keep being placed in order for DNA polymerase to build. These fragments that result are known as Okazaki fragments. Primers have to get replaced with DNA bases since the primers were made of RNA. Ligase, the gluing enzyme, as I like to nickname it, has to take care of the gaps between Okazaki fragments, sealing them together. At the end of replicating, you have 2 identical double helix DNA molecules from your one original double helix DNA molecule. We call it semi-conservative because the 2 copies each contain one old original strand and one newly made one.
And while we focus on a single replication fork, it's important to point out that in a eukaryotic chromosome in particular, where the genome is large, the big solution to the time size limitation is having multiple origins of replication, many of these replication bubbles that will ultimately fuse together. But herein lies another problem. Prokaryotes have a single circular DNA, and so it can be copied in full and completely end where it began, the entire genome transcribed. Eukaryotes, however, have non-looped chromosomes. If you unwound the DNA, it'd be one long line. And the DNA polymerase cannot add nucleotides where it is attached. So when we end replication, we have a problem. There's no way to complete the 5' ends because where the RNA primer falls off, there's no new primer for DNA polymerase to attach and fill in the gap, meaning that repeated rounds of replication will produce shorter and shorter and shorter DNA molecules. Nature found a way to fix this, something known as telomeres. These are special nucleotide sequences at the very end of the chromosomes that don't code for any critical genes. And so while they are shortened, they postpone the erosion of the critical genes near the ends. A special enzyme called telomerase adds telomeres in our germ cells. And the length of the telomeres determines how many times that cell can be duplicated, which gives us the average number of about 40 to 50 replications per cell before the telomere is completely eroded. It has been proposed that the shortening of telomeres is connected to aging. What if we could turn on telomerase in adults and continue to replenish our cells? You'd get older annually, but would be like Tolkien's elves, unaging on the outside. It's an interesting area of science that is being explored.